Hello everybody and welcome to the webinar Impact of Standardized Flow Cytometry Protocols on the Diagnostic Accuracy of Acute Leukemias. My name is Irene Gonzalez and I am the US and Canada Key Account Manager and Support Manager for Cytognos. And it's my pleasure to introduce our keynote speaker for today, Dr. Rafik Terra from Maisonneuve-Rosemont Hospital in Montreal, Canada. Rafik Terra holds a PhD in immunology and biomedical science. He is a scientific director and quality lab manager of the flow cytometry lab in the hematology department of the Maisonneuve-Rosemont Hospital. Additionally, thanks to his extensive experience in clinical flow cytometry, he works as a teacher and guest lecturer for a number of hospitals in Canada and for the University of Montreal, as well as a consultant for different companies. A very welcome to Dr. Rafik Terra, and thank you so much for being with us today.
Thank you, Irene, for this introduction. And I would like also to thank Cytognos for this invitation and giving me the opportunity today to talk to you about the evaluation of the impact of standardized 8-color flow cytometry protocols by EuroFlow on the diagnostic accuracy of acute leukemias, and also about our 7 years of experience with the application of this standardized approach. First, I would like to give you an overview on our hospital, Hôpital Maisonneuve-Rosemont, located in a nice city, Montreal. And since its opening in 1954, today we find the research center, a center of excellence for cellular therapy, and also the Leukemia Cell Bank of Quebec, and most recently an opening of the University Institute in Hemato-Oncology. And in order to meet the requirements and the importance of the hematopoietic cell transplantation unity, Hôpital Maisonneuve-Rosemont has decided to improve the quality of its laboratories, including the flow cytometry lab. For that, we had decided to have a laboratory that meets the requirements of international recommendations from the pre-analytic phase until post-analytic phase, including a structure composed by different levels of decisions with the technologists, specialists, and physicians. In parallel, we had decided to apply a standardized approach of flow cytometry, which is already available at this time, which is the EuroFlow approach. The EuroFlow consortium promotes standardization of flow cytometry immunophenotyping, evaluation of new antibodies based on their experiences and on the WHO classification, define protocols and comprehensive panels, create also a novel software which is InfiniSight, and they developed also a large database linked to this software. So actually we can notice the importance of the flow cytometry on the diagnosis of hematological neoplasia by only the presence of a picture of dot plot on the cover page of WHO classification. And many hematological diseases have an immunophenotype so characteristic that is essential or nearly essential for the diagnosis as mentioned by the WHO classification. How flow cytometry can contribute to the classification of acute leukemia. So flow cytometry can characterize the nature of the blasts of each category, if the blasts are undifferentiated, or if they have a myeloid nature or lymphoid nature, or even a mixture of phenotype, myeloid and lymphoid. So if we look at this classification, we can see several subclasses and categories. The first one is acute myeloid leukemia based on genetic mutations and dysplasia. And then we have a category of acute myeloid leukemia not otherwise specified, which doesn't meet the previous criteria of genetic mutations and dysplasia. So we have acute myeloid leukemia with the minimal differentiation based on alphabet classification AML M0, acute myeloid leukemia without maturation and with maturation, another category of acute myeloid leukemia with monocytic component. We can see also the pure erythroid leukemia, the acute megakaryoblast leukemia, and the acute basophilic leukemia. And just notice that the category of acute leukemia based on dysplasia, which is acute myeloid leukemia with myelodysplasia related changes, this category have a blasts with an immunophenotype of any of the AML not otherwise specified. Next category is a rare and aggressive disease, which is blastic plasmastoid dendritic cell neoplasm, in which flow cytometry can play an important role if we have a bone marrow and peripheral blood manifestation. So this class was previously classified under the AML group, and now in the last version of WHO classification, it's under its own category. Then we have acute leukemia of ambiguous lineage, including acute and differentiated leukemia, and also a mixed phenotype acute leukemia. Finally, we have precursor lymphoid neoplasms, including B lymphoblastic leukemia, T lymphoblastic leukemia, and NK lymphoblastic leukemia. We can see also a new category under the TALL, which is the early T cell precursor lymphoblastic leukemia, ETP-ALL, described in the past only in the literature and actually included in the WHO classification, probably because of its possible worse prognosis compared to the other subclasses of TALL and also to the different therapeutic management of patients. In order to better understand this classification and how to integrate it to hematopoietic process, we'll briefly describe the differentiation of hematopoietic cells. So hematopoiesis is a remarkably dynamic process, and the marrow normally produces approximately 100 billion cells per day. Around 200 billion red cells, 100 billion platelets, and until 60 billion neutrophils. Numbers that are just sufficient to match the rate of peripheral destruction of these formed elements. So the hierarchical model you can see here of hematopoiesis places the hematopoietic stem cells, the HSC, at the root of this tree composed of a series of multipotent, oligopotent, and unipotent progenitors that finally differentiate toward mature hematopoietic cells in the bone marrow, except for T cells which have differentiation into the thymus instead of the bone marrow. So the HSC are essential for the maintenance of hematopoiesis and are by definition multipotent, meaning that they can give rise to all other hematopoietic cells. So when the HSC divide under homeostatic conditions, it's thought that at least one of the two daughter cells remains in HSC, which is a critical property referred to as a self-renewal that maintains the HSC numbers. So a multipotent progenitor cells, the MPP, the immediate progeny of the HSC, have a full lineage potential but a limited self-renewal ability. So the MPP give rise to oligopotent progenitors then, the common myeloid progenitors, CMP, and the multilymphoid progenitors, the MLP. So the CMP differentiate into the GMP, the granulocyte macrophage progenitors, and the MEP, the megakaryocyte erythroid progenitors, which then give rise to unilineage progenitors and mature cells, which are erythrocytes and megakaryocytes and platelets. You can see also that the CMP differentiate also to classical dendritic cells, which are myeloid dendritic cells, and also to plasmastoid dendritic cells. The MLP, which are the multi-lymphoid progenitors, are able to generate both the GMP and also the CLP, the common lymphoid progenitors, which differentiate into B, T, and K cells. So the CLP also can give rise to plasmastoid dendritic cells and classical dendritic cells myeloid, like the CMP. So you notice here that the plasmastoid dendritic cells can have two different origins, lymphoid and myeloid. And all this process reflects the plasticity of hematopoietic progenitors. Also, we can define the stem cells and progenitors by their immunophenotype, expressing CD34 positive and without expression here of the CD38, so CD38 negative, at the level of the multipotent progenitors, the HSC and the MPP. Then the expression of CD38 reflects the commitment of the MPP toward the oligopotent progenitors and also toward mature cell lines. Unfortunately, with the advancing age, environmental factors, and most probably because of bad luck, the hematopoietic system will undergo genetic mutations and thereby induce the leukemic stem cells. So the formation of leukemic stem cells, the LSC, in myeloid leukemia may result from mutations in cells at different stages of the hematopoietic hierarchy. In chronic phase of CML patients, the presence of BCR-ABL in all blood lineages suggests that the LSC in CML has multi-lineage differentiation capacity and is derived from an hematopoietic stem cells or an early MPP. In contrast, in blast crisis of CML or in AML patients, the LSC exhibit immunophenotypes of oligopotent progenitors such as MLP or GMP, and this supports the concept that other more differentiated cells can give rise to the leukemic stem cells after reacquisition of self-renewal capacity, further supporting the notion that the leukemic stem cells can have quite diverse immunophenotypes and that could be generated from CD38 positive, CD34 positive, CD38 negative, or CD34 positive, CD38 negative progenitors. So if one mutation or multiple mutations happened at the very early stage of differentiation, the leukemic stem cell give rise to cells that have lost self-renewal and cannot differentiate terminally, which are the blasts. So the acute and differentiated leukemia is the result of a blockage at this stage of hematopoietic differentiation with a multipotent progenitor's immunophenotype, which is CD34 positive and CD38 negative without any commitment. If the blockage happened at the lymphoid stages of differentiation, we can also attribute to blastic plasmastoid dendritic cell neoplasma, CD56 positive, the myeloid origin if the origin of the blockage is the common myeloid progenitors. We know also that the plasmastoid dendritic cells, CD56 negative, can be mixed with the monocytic blasts or myeloid blasts, suggesting a myeloid origin for some cases of acute leukemia with a proportion of these plasmastoid dendritic cells. The other myeloid leukemias are the result of a blockage at a different stage of differentiation of monocytic, granulocytic, erythroid, or megakaryocytic cell line. So acute leukemia is a heterogeneous group of diseases with a poorly differentiated and more differentiated immunophenotype. Here I show all the poorly differentiated acute leukemias with the possibility of stem cell, myeloid, or lymphoid antigen expressions. like acute and differentiated leukemia, ETP-ALL with myeloid and lymphoid potential, blastic plasmacytoid dendritic cells CD56 positive, which is cytoplasmic MPO negative also, and the AML M0. And also I include AML with myelodysplasia related changes because their blast have any immunophenotype of the AML NOS. So as this poorly differentiated acute leukemia have some similarities in their immunophenotype, so confusion can occur between early immature lymphoid or plasmacytoid dendritic cells, acute leukemia, and acute myeloid leukemia with minimal differentiation AML M0 if proper immunophenotyping panels are not performed. So if we look in details at the immunophenotype of these early leukemia forms, we notice that they have almost the same immunophenotype except for a few markers like cytoplasmic CD3, which is usually partial or positive DIM at the ATP-ALL acute leukemia, or a strong expression of CD7 and CD5 DIM expression at also ATP-ALL. And we can see also the specificity of a strong expression of CD123 at the BPDCN. So we can realize that if we don't use a comprehensive panel in our routine, we can easily be confused between these entities. So we ask the question, which is, what is the impact of non-standardized laboratory developed panels on the accuracy of acute leukemia diagnosis? So to answer this question, we have decided to reanalyze cases of poorly differentiated acute leukemias applying the standardized EuroFlow approach. So the reanalyzed cases was provided by the Quebec Leukemia Cell Bank, which has specimens of patients previously diagnosed with several types of acute leukemia. So the Quebec Leukemia Cell Bank is a provincial program funded by the Cancer Research Network and other partners. And also since 2001, they have the objective to support basic and clinical blood cancer research, and they provide phycalized high-quality specimens which have been cryopreserved according to international standards. Our objectives were to establish the extensive immunophenotypic profile of selected cases. Some of them are acute myeloid leukemia with minimal differentiation, and also some cases of acute myeloid leukemia with myelodysplasia related changes, which have blasts with the immunophenotype of any of the AML NOS. And then we have some cases of acute leukemia of ambiguous lineage, including undifferentiated acute leukemia and mixed phenotype acute leukemia. Then we identified cases corresponding to BPDCN and ETP-ALL, and described also the associated cytomorphology and immunologic expression pattern. Then we compared the survival data of the different groups of patients according to their immunophenotype and the treatment protocol used. And finally, we have done NGS for some cases using an AML-related gene panel. So we have selected 49 cases collected in the last 15 years, excluded also cases with monocytic and or myeloid differentiation, and also we kept some controls, about 10 cases with cytoplasmic MPO partially expressed or fully positive, without any reported evidence of monocytic component in the initial diagnosis. Also, among these cases, we had some cases with CD7 positive bright and CD56 positive. So even they are MPO positive, we wondered if they are not misdiagnosed and was not T-ALL or PBDCN. So the nomenclature of the 49 cases was reported by the BCLQ was AML with myelodysplasia related changes for 19 cases, 9 cases with AML and OS, so M0 or not classified by FAB as reported by the BCLQ, 5 cases with AML with minimal differentiation, so it's like AML M0, 1 case with AML without maturation, 4 cases with acute and differentiated leukemia, a few cases with mixed phenotype acute leukemia, B myeloid, T myeloid, BT, and NK myeloid. 2 cases with T-ALL and 2 other cases with B-ALL. About the treatment, 32 cases had induction therapy, 15 cases with palliative or supportive care, and 2 cases with a known therapy. So for our analysis, we have applied the EuroFlow acute leukemia algorithm, which include in the beginning a screening tube, unique screening tube, which is the acute leukemia orientation tube, the ALOT tube. And then based on the results obtained with this screening tube, we applied one of the characterization panels, which are BCP-ALL, T-ALL, or AML-MDS. So the ALOT tube is composed by 8 markers, each one specific for lymphoid or myeloid lineage, and also for immaturity with CD34. So if we had cytoplasmic MPO positive, so then we process with the AML-MDS panel with 7 tubes. If cytoplasmic CD79 or CD19 are positive, then we have done the BCP-ALL panel with 4 tubes. And if we have positivity of one of the surface or cytoplasmic CD3 and with CD7 bright, so we process with the T-ALL panel with 4 tubes. If no one of this marker is positive, so we process first with AML-MDS, and then if we have suspicion of plasmacytoid dendritic cells, so we have decided to confirm these results with 3 more specific markers, which are TCL1, CD303, and CD304. The ALOT tube is able to detect until 80% of acute leukemia, even without the use of the characterization panel. So its evaluation with the InfiniSight software, as you can see here, linked to the database of more than 400 patients with different acute leukemia, which include T-ALL, B-ALL, AML, and ambiguous acute leukemia, including acute and differentiated leukemia and a few cases of mixed acute leukemia, allowed to confirm the strength of the ALOT tube for its ability to discriminate between the 3 types of acute leukemia, AML, B-ALL, and T-ALL. So if you look at the pairwise analysis of the 3 categories of acute leukemia, you can see that the BCP-ALL, here in blue, is clearly separated from the AML in red and T-ALL in green. Conversely, T-ALL and AML groups here showed a slight overlap, and that is due to some cases mostly corresponded to immature T-ALL that fit the criteria for ETP-ALL with unusually dim cytoplasmic CD3 expression and immature CD34 positive cytoplasmic MPO negative and CD7 positive AML, as it can be reported for AML M0. So this overlap may be the consequence for both the biological nature of immature blast cells in some malignancies arrested at the stage of early T myeloid development and the high phenotypic heterogeneity of non-lymphoid acute leukemia cases. So the Infinity software is able also to evaluate what is the most informative, the 5 most informative markers for the best discrimination between each diagnostic entity. For example, if you use in your routine a screening tube or a panel without, for example, a cytoplasmic MPO, without CD7 and without cytoplasmic CD3, you will not be able to easily discriminate and to detect the T-ALL entity and well discriminate between AML and T-ALL. Then if we look in more details to the AML MDS tube, we can see that it's composed by 7 tubes with a backbone here, HLA, DR, CD45, CD34 and CD117. So this backbone with these markers repeated in each tube allow us to analyze all the tubes in the same time. Then we have each tube for a specific lineage. For example, the tube 1 is for neutrophilic lineage, the tube 2 is for monocytic lineage, the tube 3 is for erythroid lineage, the tube 4 is for some aberrant expression of lymphoid associated markers and abnormal lymphoid maturation. We can see also the CD56 which is expressed on blastic plasmastoid dendritic cells neoplasma. The tube 5 also for aberrant expression of markers and detection of stem cells and also for correlation with some genetic aberration. The tube 6 for diagnosis of blastic plasmastoid dendritic cells neoplasma, also focused on megakaryocytic cells, also for basophilic cells. And the tube 7, it's for mastocytosis and characterization of AML-M7. So with the 33 antibodies inside the AML-MDS panel and the advantage of the use of the Infinity software, we can analyze all the different population inside the bone marrow with the automatic population separator configuration, the APS here, as you can see. So we can easily separate each population, and if you look at the normal bone marrow, we have here in green the granulocyte precursors and neutrophils. In red we have stem cells. In blue we have monocytes and precursors, and in orange we have the lymphocytes. And also the use of the two markers CD117 and HLA-DR is helpful to visualize the maturation of the population. For example, here in red you have the stem cells which differentiate into monocytes in blue by losing the expression of CD117 and acquiring the expression of HLA-DR. And the maturation also of the stem cells in red toward the granulocytes by losing the expression of CD117 and also for HLA-DR. We can also depict the relative distribution and phenotypic profile of CD34 positive hematopoietic stem cells in a representative bone marrow sample. Here we have a normal bone marrow, including both in committed in the black dots, CD34 positive, CD38 negative, and committed precursors to the B and T and K lymphoid lineages in pink, pink dots here, and to the different myeloid cell lineages in only single automatic population separator diagram, which is APS1 here. So at the right of this figure we have a representation of immunophenotypic patterns of distinct hematopoietic cell compartments, showing here the maturation of neutrophil by using the tube 1 of the AML-MDS panel, the monocytes by using the tube 2, and erythrocytes by using the tube tree. The arrows also represent the maturation pathway from immature CD34 positive hematopoietic progenitor and precursor cells in red and orange dots to maturing and mature cells for each cell line which are CD34 negative. So if we look at the neutrophil maturation, we can see that the progenitors CD34 positive already express CD13. They increase a little bit this expression, then they start to lose the expression of CD13, and they will increase and start to express CD11b and increase its expression until it reaches the last step of maturation, which are neutrophils CD11b bright and CD13 bright. For the monocytes, the same thing. So from the progenitors CD34 positive, we have expression first of CD14 until the maximum expression of this marker. Then the monocyte precursors start to express CD300E, which is RM2, until reaching the last step of maturation, which are mature monocytes, CD14 positive and CD300E positive. And the color lines here represent the level of expression of markers corresponding to each maturation stage for each cell lineage. So the AML-MDS panel is not allowing us only to report if markers are positive or negative, but also allow us to analyze the maturation of each cell line and to detect in our samples all the abnormalities due to the dysplasia or mutations. And also with this panel we can assign a nature and a stage of maturation blockage of our blasts. For our study, all the events were acquired on BD FACSCON22 instrument, and data was analyzed using Infinisight software manually and also by applying them on database, and also cytomorphologic examinations of the identified samples were reviewed. First, we evaluated the impact of cryopreservation and thawing process on immunophenotyping in order to confirm the high quality of our specimens and that our specimens maintain a similar immunophenotype compared to fresh cells. So flow cytometry was performed on the same sample before on the sample A and after cryopreservation and thawing 20 months later on the sample B. And you can see that all major cell populations with the different colors are easily identified and associated with the identical fluorescence intensity in both analyses. Also, the profile maturation patterns here with the arrow remains intact as you can see on monocytes in blue by expression first of CD64 and then by the expression of CD14. Also on granulocytes by expression of CD13 and CD11b, and also as you can see by the expression of CD117 and HLIDR on stem cells and monocyte and granulocyte precursors. We also confirm the quality of our specimens by evaluating the median fluorescence intensity of different markers. So this graphic represents the median of different markers on monocytes and blasts for fresh and cryopreserved cells. And as you can see, that the intensity of marker expressions is not at all affected. So our results show that among the 49 cases reanalyzed, we got 18 cases modified, 5 cases refined, and 26 cases without any change. The refined cases were those without any impact on treatment. So we refined 4 B-ALL, including 2 pre-B-ALL, 1 common B-ALL, and 1 pro-B-ALL, and also 1 case of T-ALL to a cortical subclass. Concerning modified cases, these were cases with revised diagnosis and or with significant additional finding, with a possible impact on treatment of some of them. So among these 18 modified cases, we got 1 megakaryoblast leukemia, 3 early T cell precursor acute leukemia, which is ETP-ALL category, 2 cases of typical blastic plasmastoid dendritic cell neoplasm CD56 positive, 8 cases acute leukemias with different proportions of plasmastoid dendritic cell CD56 negative with or without additional monocytic component, and finally 4 cases of acute leukemia with the additional monocytic component. In this table, we detailed the clinical history of acute leukemia for patients with modified diagnosis. So we have the year of the disease, the age of each patient, the initial diagnosis as reported by BCLQ, the initial therapy with or without allogenic transplantation, the survival per days, and also the revised diagnosis based on immunophenotype by using the EuroFlow panel. The first case of acute megakaryoblast leukemia was initially reported as AML with myelodysplasia-related changes with the myeloblast, which has expression of CD56 and CD36. So it was reported as aberration or probably NK cell nature. But as we have all the markers in EuroFlow panel, we were able to confirm the megakaryocytic nature of this blast instead of reporting myeloblast with the CD56 expression and 36 expression. Then we have the 3 cases of ETP-ALL, initially reported as AML M0, and 2 others as T-ALL and mixed phenotype acute leukemia T and myeloid. And we didn't report these cases as refined cases for the simple reason that the ETP-ALL seems to have worse prognosis, and also because of the therapeutic management of the patients, which seems to be different compared to other subclasses of T-ALL. Then we have the 2 typical blastic plasmastoid dendritic cell neoplasm CD56 positive, initially reported as T-ALL AML and mixed phenotype acute leukemia NK myeloid. In 4 other cases, we reported the presence of a different proportion of plasmastoid dendritic cells, immature plasmastoid dendritic cells, CD56 negative, CD34 positive, mixed with the proportion of blasts with minimal differentiation. And initially, this acute leukemia was reported as AML with myelodysplasia-related changes with any evidence of presence of plasmastoid dendritic cells. Another case for acute and differentiated acute leukemia, and another case of AML with minimal differentiation, and finally another case with AML with myelodysplasia-related changes. 4 other cases were reported with, again, different proportion of immature plasmastoid dendritic cells, but here with mixed with the monocytic cells, and also 1 case with monocytic and pathological blast B cells. And initially, these cases were reported as AML with minimal differentiation, acute and differentiated leukemia, AML NOS not classified by FIB, and AML with myelodysplasia related changes. Finally, we got 4 cases of acute myelomonocytic leukemia because we demonstrated clearly the presence of a proportion of monoblast and promonocytes mixed with a proportion of myeloblasts or blasts with minimal differentiation. So how the EuroFlow panel can help us to detect each specific population. So I start with the early T-cell precursor ALL. So if we look at the thymus, we can see the differentiation of the T-cells from early T-cell progenitors at the corticomedullary junction until the sample positive T-cells, thymocytes, CD4 positive and CD8 positive at the medulla. So if there is a mutation at the very early stages of T-cell differentiation or oligopotent progenitors causing an accumulation at this stage of early T-cell precursor cell, so we will have the ETP leukemia with its specific and typical immunophenotype, cytoplasmic CD3 dim or partially expressed, CD7 positive bright, CD2 described in the literature as negative, but in our routine is more often positive than negative. Also, the typical dim expression of CD5 in the literature reported as less than 75% of positivity. We have also one or more myeloid markers, CD13, CD11b, and CD117. So if in your routine panels of diagnosis you don't have these markers which can help you to detect the ETP entity, which are CD5, CD117, CD33, CD13. Also, you can add CD11b and CD2. It will be very difficult to characterize the ETP-ALL. When we apply the 3 cases of ETP-ALL on the database, all the events in red here fall down inside the 2 standard deviation curves for the T-ALL group, here in blue, because the 3 cases express cytoplasmic CD3, CD7 bright and CD34 positive, and don't express cytoplasmic MPO, CD19 and cytoplasmic CD79. So it's why we have clearly this assigned T cell category. But if you look in more details, you can see that these 3 red dots, which are our case, fall inside the blue category at different position. On the top for the first case, in the middle for the second case, and in the bottom for the third case. And that is probably because of the different intensity expression of all the positive markers inside the allotube. And that is the reason why the compass in the bottom here doesn't show exactly the same orientation of the arrow. For example, for the case 04104, as the red dot here is in the middle of the T-ALL group, so the arrow with the compass is pointed in a unique main group, which is T lineage at 88%. For the 2 other groups, we can see that the dot is at the limit of the standard deviation 1 and inside the standard deviation 2. So it's why there is a dominant group, which is T cells, and another group with the transition, which is B or myeloid, dependent of the case. Then to be able to detect plasmacytoid dendritic cells, CD56 positive or CD56 negative, we have used the AML-MDS panel, in which the tube 6 contains 3 markers expressed on the plasmacytoid dendritic cells, which are HLIDR, CD123, and CD4. We have also this CD56 marker in tube 4, and we can also find other markers expressed on plasmacytoid dendritic cells CD56 positive and even CD56 negative, which are CD36, TDT, CD7, NG2, and even some myeloid markers CD13 and CD33. So if we have suspicion of the presence of plasmacytoid dendritic cells, we have decided to use 3 more markers, specific markers for plasmacytoid dendritic cells, which are cytoplasmic TCL1, CD303, and CD304. So here we have 2 cases, one from the typical PBDCN with the plasmacytoid dendritic cells CD56 positive, and the second one PBDCN CD56 negative. So if we look at the first group, CD56 positive, so the plasmacytoid dendritic cell population in red is CD45 dim. It's CD34 negative, HLIDR positive, CD7 positive, TDT positive, 36 here is expressed also. We have also a specific CD123 bright. CD304 also is positive here. CD4 also is positive in the first case. And we have a bright TCL1 here for the PBDCN CD56 positive. The second case, CD56 negative plasmacytoid dendritic cells. We still have these cells at CD45 dim region, but mixed with the other blast population or a high proportion of monocytic cells. So these PBDC CD56 negative are usually CD34 positive, HLIDR positive. Here we have CD7 partially expressed and TDT also. Can express CD33, express CD123 even brighter than the PBDC CD56 positive, express also CD304 and CD303. So if you have almost these markers, it will be easy to target and to characterize these 2. So as the CD123 marker seems to be brighter in the plasmacytoid dendritic cell CD56 negative compared to plasmacytoid dendritic cells CD56 positive. So for the first time, we report that this marker could be useful to identify and discriminate between these 2 populations. Now, how we can detect the monocytic population with the EuroFlow panel? By the use of the second tube of AML-MDS panel, we can characterize the monocytic population. So here we have the monocytic maturation patterns in normal bone marrow in A and in leukemic bone marrow in B. So in normal bone marrow, if you look at the arrow in yellow here, so this arrow shows the expected maturation for monocytes by the loss of CD117 from the stem cell until the mature monocyte. Then we have expression or increased expression of CD64 followed by the expression of CD14 and finally by the expression of CD300. So the mature normal monocytes express CD64 bright, CD14 bright, and CD300 positive with HLA-DR positive. In B, we have two cases of myelomonocytic leukemia. The case 1 has a normal maturation pattern, but you can see the abnormal accumulation in red of blast cells and of promonocytes in light blue and in dark blue of monocytes. In case 2, another myelomonocytic leukemia case, in which the maturation pattern is abnormal, based on the expression first of CD300 and then on CD14. This abnormal pattern suggests a mutation of NPM1. Here we have two cases, initially reported without a high proportion of monocytic cells. In the case 06, as you can see, there is no clear evidence by morphology and cytochemistry, and initially it was reported less than 10% of monocytes. In contrast, when we used the EuroFlow panel, we can recognize the maturation profile of monocytes with this high proportion, clearly, of monocytic cells evaluated at 46% of monocytic component. The case 2, which is the 05022, also no really clear evidence of monocytic cells by morphology and cytochemistry. In contrast, with the tube 2 of EuroFlow panel, we can recognize also the pattern of monocytes, perhaps with the expression of dim HLA-DR, which could happen in some cases when there are some mutations. And also we have the pattern of CD64 bright and the expression of CD300 at mature monocytes. And in this case, the monocytic component was evaluated at 44%. By using the database and the COMPASS, so the two populations for the two cases, monocytes and myeloblasts, fall into the category of AML, here in red, and the arrow on the COMPASS pointed toward the myeloid lineage at more than 90%. For megakaryoblast leukemia, by using the tube 6 and 7 of the AML-MDS panel, we have clearly demonstrated the expression of a specific marker of megakaryocytes, which are CD36, also CD42A, CD61, CD41 and CD42B. And by morphology, we have done a cytospin post-thawing by doing cytochemistry also to confirm that we don't have a non-specific staining, and to also confirm the quality of the cells we have stained. We also performed next generation sequencing in this case. We repeated the staining, the immunophenotype on two different samples cryopreserved, just to confirm these results. And then we have done next generation sequencing to see or to detect potential pathogenic mutation reported in the literature, TED2 or KMT2C or RUNX1 or GATA1. And finally, we have found that GATA1 transcription factor is mutated in this case, and GATA1 has interestingly been reported in acute megakaryoblast leukemia. So in conclusion, these results support the major role of a comprehensive immunophenotyping panel in the diagnostic accuracy of acute leukemia by multicolor flow cytometry. We demonstrated that incorrect diagnosis may result if we use limited immunophenotyping panels, even with the global characterization including morphology and cytochemistry. And extensive immunophenotyping using EuroFlow approach can refine the diagnosis with cases of ETP-ALL by using specific markers, and PBDCN also if we have the necessary markers to detect this category of cells. And also, which was unexpected, cases with significant monocytic component, demonstrating that flow cytometry, the immunophenotype, is more sensitive than morphology or cytochemistry. So the use of standardized approach of flow cytometry can lead to more precise diagnosis of poor prognosis leukemia subtypes like ETP-ALL and PBDCN, and has also the potential to change the therapeutic approach with the potential improvement of clinical outcome.